This presentation is the first of 6 parts or 6 brief presentations on intracellular membranes. We'll be looking at cellular organelles and vesicular traffic. And the learning objectives, and these objectives will basically apply to all of the 6 presentations. You should be able to describe various protein traffic pathways in eukaryotic cells, be able to describe the events associated with protein translocation and glycosylation in the ER. You should be able to identify which organelles belong to the membrane flow family. We talked about membrane flow earlier, so we're going to come back to that now. You should be able to describe functional compartmentalization of the Golgi and how molecules move through the Golgi. You need to be able to differentiate between constitutive and regulated secretion. And then finally, be able to describe how lysosomes are formed and how lysosomes relate to intracellular digestion. Now, your learning objectives for the laboratory: be able to describe how intracellular compartments are related to the function of the pancreatic acinar cell and liver hepatocyte. And this is to kind of put the cell biology into perspective. And then begin to be able to describe the histological organization of the pancreas and the liver. This objective is not so critical this early in the course, but it will build, and so beginning to appreciate some of this histology will make what you do a little bit later in the course easier. Now, there are a variety of key concepts that I'd like you to keep in mind as we discuss the ER and the Golgi. First of all, these organelles are involved in a variety of metabolic functions in the cell. When we talk about protein traffic through these organelles, we're going to be talking about membrane flow. When we talk about protein glycosylation, we're talking about membrane fluidity. When we talk about lipid traffic through these organelles, we're talking about membrane flow. We will spend a little bit of time reviewing and going over the signal hypothesis as that relates to protein translocation. And everything that we say in these presentations is absolutely dependent on an intact cytoskeleton. We won't talk too much about the cytoskeleton in these presentations, but that will come later in the course. Now, I'd like to start with this diagram and look at membrane flow, and I want to talk about vesicle budding and fusion during transport. So this is a hypothetical donor compartment in a cell, and you can see these black lollipop structures would represent membrane proteins, and the little red dots would represent some cargo that's in this particular compartment of a cell. And notice the orientation of the lipid bilayer. Now, what happens, let's suppose that you want to transport these red components from one area of the cell to another area of the cell. Well, a vesicle is going to bud off, trapping some of this cargo, and this vesicle is going to fuse with a target compartment. So it's moving the cargo from a donor compartment to a target compartment. And as that is occurring, coincidentally, some of these membrane protein components are also moving from the donor compartment to the target compartment. Now, that seems pretty obvious, but it's something that's worth just keeping in mind. So now, as membrane movements occur, these membrane movements might occur as small vesicles, as shown in this example with a vesicle budding off. They could move as a membrane tube, or an entire cisternae may move, and that will become clear as we talk about the Golgi in a little bit. Now, the cartoon emphasizes that soluble components, represented by these little red dots, are moving from lumen to lumen. The cartoon also suggests that membrane proteins and membrane lipids are also transferred from one compartment to another. And something that should be obvious, but we tend not to think about it that much, is the membrane topology is maintained. So for example, in this compartment, the one face of the lipid bilayer is shown in blue, another face is shown in green. As you follow this membrane movement through the vesicles from the donor compartment to the target compartment, the membrane topology is maintained because you can see the blue face of the bilayer stays on the outside. The blue face of the bilayer here stays on the outside. The blue face of the bilayer finally here is on the outside. So membrane topology is maintained, and that's also an important concept. Now we also talk about cell polarization, and this can have basically two meanings. The first would be, if we look at this as a hypothetical, say, epithelium, like maybe in the GI tract where it's an absorptive epithelium, we can talk about an apical surface, and in the diagram this apical surface is shown elaborated into microvilli, and we can talk about a basal and a lateral surface, or sometimes termed basolateral surface. So we can say this cell is polarized in that there's a distinct apical membrane domain which is different from the basolateral membrane domain. We can also talk about a cell being polarized with respect to how the organelles are arrayed. So if this was a secretory cell, for example, you would tend to have the rough endoplasmic reticulum located maybe towards the basal region of the cell with a Golgi apparatus, and then secretory vesicles budding off of the Golgi apparatus. So we might say this cell is polarized for secretion from the basal region through to the apical region. And indeed, when you look at histological staining with stains like H&E of secretory cells, you can get a sense of that distinct polarization for secretion. And we'll come back and talk about that a little bit later. Now, here is a table, and it's just looking at relative volumes occupied by various intracellular compartments in a typical cell. And here we're considering the typical cell as a liver cell, the hepatocyte. First of all, if we look at the different compartments and look at percent of cell volume, we can say, well, obviously the cytosol has the largest percent of cell volume. And by cytosol here, we're meaning the cytoplasm of the cell, but not counting the membrane-bound organelles. So cytosol is the biggest volume compartment, followed by mitochondria. But then in today's discussion, we're going to be talking about the cisternae of the rough ER, smooth ER, and Golgi, which is about 15% of the volume. And then we'll talk about peroxisomes, lysosomes, and endosomes, another 3% of the volume. So together we're talking about almost 20% of the cell volume when we talk about these intracellular membrane-bound organelle compartments, excluding the mitochondria, obviously, and excluding the nucleus, obviously. Now, another thing I'd like to help you to understand is as we compare different cell types. So let's look at the hepatocyte, which is a prototypical cell, and the pancreatic acinar cell, another prototypical cell. And we can look at distributions of membrane, particularly the rough ER, smooth ER, and Golgi membranes in these cells. Now, first of all, let's consider the hepatocyte. As you will recall from biochemistry, the hepatocyte does just about everything in the body from the perspective of biochemical properties. So it synthesizes proteins that are going to be part of, say, serum. Hepatocytes also detoxify lots of substances. So you'd expect reasonable amounts of rough ER, smooth ER, and Golgi. But then compare the hepatocyte to a pancreatic acinar cell. The pancreatic acinar cell is highly specialized for secretion for exocytosis, for proteins that are going to be excreted from the cell. In such a cell, there's almost twice as much rough ER as in the hepatocyte. So a cell highly specialized for protein secretion has way more rough ER than a typical cell. On the other hand, the acinar cell is not so involved in lipid biosynthesis, not so involved in detoxification of xenobiotic substances, so has very little smooth ER compared to the hepatocyte, which is involved in lipid biosynthesis and in detoxification of a lot of foreign substances. So the distribution of rough ER and smooth ER to a great extent reflects the metabolic activity of a cell. And we will ask you in the laboratory over the next few days to begin to look at hepatocytes and pancreatic acinar cells and correlate these cells to the cell biological and biochemical functions in relation to the distribution of the various membrane-bound organelles. I would like to remind you of just some topological relationships between the various compartments in eukaryotic cells, and we can look at these compartments vis-a-vis exocytosis or secretion and endocytosis or bringing material into the cell. Now, first of all, what this cartoon shows is what's colored orange would be extracellular space. And what this cartoon emphasizes for you is that the luminal compartment of organelles like the ER, or the luminal or cisternal compartments of the various stacks of the Golgi, are spaces that are equivalent to the extracellular space. Same thing for secretory vesicles. The luminal compartment of the vesicles is equivalent to the extracellular space. This perhaps makes more sense if you think of an endocytic vesicle, something pinching in, as shown here, a vesicle pinching in, bringing something from the outside of the cell into the cell. This compartment would be equivalent to extracellular space. So right here where I've had my cursor, this is indicating secretion, something budding from the Golgi going to a vesicle and the material in this vesicle being released to the outside of the cell. Here, a vesicle pinching off, and the contents of this vesicle would be equivalent to extracellular space. So, a few quick words about the ER and the Golgi. If we look at the ER is a single membrane-bound compartment. About 50% of the total cell membrane is contributed to by the ER. If we talk about regions of the ER that have ribosomes bound to them, we're talking about rough endoplasmic reticulum. This region of the ER specializes in translation, that is, synthesis of proteins. And these proteins will be either proteins that remain in membranes, proteins that are secreted from cells, or proteins that are destined to go to other organelles. On the other hand, if we talk about regions of the ER that don't have ribosomes bound to the membrane, we're talking about smooth ER, and regions of the smooth ER are specialized for lipid biosynthesis, for detoxification of xenobiotic or foreign substances, and the smooth ER, especially in muscle cells, is a major storage site for intracellular calcium. Take-home message: the ER is a single membrane-bound compartment. Contrast that to the Golgi. The Golgi is a series of single membrane-bound compartments that are stacked, and it's almost like each stack in a Golgi is a different membrane-bound compartment. We'll talk about that in a little more detail as we talk about the Golgi. Biochemically, the Golgi receives proteins and lipids from the ER, and it receives this cargo by vesicles. It routes this cargo to other parts of the cell by vesicle movement. A major function of the Golgi is to covalently modify material that comes to the Golgi from the ER. We're also going to talk about a fairly significant amount of recycling between the ER and the Golgi. We'll refer to a compartment called the our Golgi intermediate compartment, and we'll also talk about a compartment called the endosome compartment, which functions primarily in endocytosis, and we'll talk about the endosome compartment a little bit later. This is a slide that's 50, almost 60 years old now, and it shows our traditional understanding of relationships between the ER and the Golgi. Here, showing the ER, this would be rough ER with ribosomes bound to it, and you might imagine these ribosomes are synthesizing protein. You might imagine this protein is getting transported to the Golgi, and then this protein might be being modified through the Golgi and transported through the Golgi, and then vesicles might bud off of the Golgi, and these vesicles might bud off in a signal-mediated manner for a secretion. This would be called regulated secretion. Maybe the cell is receiving a signal to secrete, and these vesicles would fuse with the plasma membrane and secrete their substance. These vesicles might bud off from the Golgi and be routed to lysosomes. We'll see a little bit later that this is also in a signal-mediated manner. Or alternatively, you might have vesicles that are budding off of the Golgi and that are constantly fusing with the plasma membrane in a constitutive manner, delivering material from the Golgi to the outside of the cell. This would be called constitutive secretion, and this type of secretion would not require any specialized signal. This part of the cartoon is looking at endocytic retrieval of membrane, and 50 or 60 years ago we thought that these vesicles would bud off and go straight to the Golgi. We now know that this endocytic retrieval involves a totally new compartment that's been described maybe over the past 10 or 15 years called the endosome compartment, and we'll talk about that a little bit later. So here is a kind of roadmap, if you want, to look at movement of material within the cell. And we can talk about movement from the cytosol to the nucleus, and this is a two-way movement as material will go through pores in the nuclear envelope, so we'll call that a gated type of transport. We're going to spend considerable amount of time talking about transmembrane transport, where material goes from the cytosol directly across a membrane into the endoplasmic reticulum. We will talk about that in great detail. We're not going to talk at all about movement that way in mitochondria, and clearly not in plastids, which would be for plant cells. But you see this type of movement across membranes to the ER, to mitochondria, to peroxisomes, and if you're a plant cell, into organelles like chloroplasts. We're also going to talk about vesicular transport, that is movement from the ER to the Golgi, to secretory vesicles, to lysosomes, to the cell surface. This is by vesicle transport, and this is by and large a two-way street because vesicles can move in both directions. So vesicles might move from the cell surface to endosomes, back through to the Golgi, and potentially to the ER, and vesicles will move from the Golgi to endosomes, to lysosomes, to secretory vesicles for cell secretion. So very much a two-way street when we talk about vesicular transport. Finally, this is a slide that we showed you earlier. I now want to put into perspective some concepts of membrane flow, exocytosis, and endocytosis. And we can talk about exocytosis broadly as part of the biosynthetic pathway, and we can talk about endocytosis broadly as part of the phagocytic pathway. And we're going to talk about this in relation to a regulated pathway and a default pathway. So we would talk about the default pathway as a pathway of constitutive secretion. And what this is implying is material that's membrane bound is moving from the ER to the Golgi, through the Golgi to secretory vesicles, and the secretory vesicles are moving towards and fusing with the plasma membrane to release substance. This is membrane flow. It's constitutive secretion because there's no signal that's required for this movement to occur. We can broadly refer to constitutive secretion as part of the default pathway. On the other hand, we might talk about regulated secretion, again, material moving from the ER to the Golgi, through the Golgi, perhaps as vesicles or perhaps as entire cisternal stacks moving, as we'll talk about, vesicles budding off. These vesicles stay within the cytosol and don't fuse with the plasma membrane for secretion unless there is a specific signal to tell the vesicles to fuse and secrete. We can talk about this as signal-mediated secretion as part of a regulated pathway. In a similar manner, we can talk about signal-mediated movement of vesicles to lysosomes. So once again, movement from ER to Golgi, through Golgi, and the vesicle going to a lysosome, again under a specific signal. So signal-mediated. We can talk about a phagocytic pathway, material being brought in from the plasma membrane, a vesicle forming. The vesicle would fuse in an early endosome compartment, go to a late endosome, and eventually to a lysosome. Now, this phagocytic pathway can be somewhat complex because we can talk about endocytosis, which does not require any signal. So we might talk about something like a fluid-phased pinocytosis, where you're just bringing in small amounts of fluid. Or we might talk about signal-mediated endocytosis, as we alluded to in an earlier presentation on receptor-mediated endocytosis. We might also talk about a recycling pathway where you could recycle material from endosomes back to the Golgi, through the Golgi back to the ER, and we will talk about this in fair detail a little bit later. We might also talk about a recycle pathway at the cell surface, because maybe a vesicle is endocytosed, as we talked about with LDL internalization. Part of the material is routed through early and late endosomes to lysosomes for digestion, like for digestion of the LDL particles, so the cell could utilize cholesterol. But at the same time, components of this membrane that, say, carry the receptor might be recycled to the cell surface. So the recycle pathway can be a complicated pathway. So we've talked about a regulated pathway and a default pathway. Now sometimes you see a term, I don't like this term, it's called curl, C-U-R-L, compartment that uncouples receptor from ligand. You might think of that compartment as the early endosome compartment. I prefer not to use the term curl, but I've seen this term in some textbooks and in some board review books. So if you see that term, I want you to equate that with the early endosome compartment, where you're uncoupling receptors from ligands. You're recycling the receptor back to the cell surface. You might be cycling the ligand through the lysosomal pathway for digestion. All of what we're talking about here is absolutely dependent on an intact cytoskeleton. We'll allude to this a little bit later.